In today's webinar, we will show you how to access and publish genetic data with SBDI. So the core of SBDI is the BioAtlas. And the BioAtlas is a web portal based on the Living Atlases platform that gathers species observation data from a range of data sources. So in the Swedish BioAtlas we have, for instance, natural history collection data, citizen science data, systematic monitoring data, and biotelemetry data. But we also have molecular biodiversity data. And by this we primarily mean metabarcoding data, but in the future we will also have metagenomics data. And most of you are probably well familiar with metabarcoding data, but for those of you that don't know what this is, this slide will kind of show you how it in principle works. So the investigator has some type of environmental sample, and from this sample DNA is extracted from the organisms or just free DNA in the sample, and then from this DNA taxonomic marker genes are PCR amplified using primers that cover these genes in more or less all of the species in the sample. And then this pool of PCR products is sequenced using a high throughput sequencing machine, so that maybe thousands or up to millions of barcode sequences are obtained for each sample. Since most of the modern sequencers are generating some sequencing errors, a sophisticated algorithm is usually applied to remove these errors, and then the resulting data is a set of amplicon sequence variants, also called ASVs, and these are then more or less error-free sequences, and these sequences are usually taxonomically annotated by comparing them to sequences in reference sequence databases. And once that has been done, one can generate a table of the different ASVs, their taxonomy, and then their counts in the sample. Often several samples are sequenced, and then you get the counts in all of the different samples. So a metabarcoding is a widely popular approach already, but it increases steadily also in popularity. So last year about 10,000 studies were published with metabarcoding, and these include from 1 up to thousands of samples. So it's a huge number of samples that are being sequenced using this method every year. And how do we then handle this type of data in SBDI? So this will show you the data flow. First, the investigator gets sequences from his or her samples. These are usually provided by a sequencing center like the National Genomics Infrastructure at SciLife Lab, for example. And then these raw sequences that are delivered should be deposited in a sequence repository such as the ENA. And we provide you with a pretty detailed guide for how to do this. So Maria will explain this later. And then the investigator denoises the sequences and generates these ASVs using, for example, the NF-core AmpliSec pipeline that we have co-developed, and Daniel Lundin will tell you about this shortly. And then when you have your ASVs, these can then be submitted to the SBDI. And then these are submitted and first are ingested in the Swedish ASV database, the ASVs together with the counts and samples and contextual data. And they will be annotated taxonomically, so that all the data inside the BioAtlas and the Swedish ASV database will have a kind of uniform taxonomy. And then the data is ingested in the BioAtlas, so that the occurrences in samples are then part of the BioAtlas. And if the investigator opts to do so, the sequences will also automatically be exported to GBIF. And then data that has been published in the BioAtlas can be searched by either using BLAST or by doing taxonomic searching using the ASV portal, and the occurrences can be retrieved from the bioatlas. And Maria Prager, who has developed this functionality, will tell you more about this later today. So basically, behind the scene, we have the bioatlas that is built on the Living Atlases platform. It has some functionality for metabarcoding data, but it's rather limited. So to be able to handle metabarcoding data in a very efficient way, we decided to develop the ASV portal and the ASV database, and these are then kind of a plug-in module to the bioatlas. And maybe you ask, why do we care about these ASVs? Why don't we just take the raw sequences and map them to reference databases and just get counts per species or taxonomic group? Well, there are some different reasons why. One is that the ASVs provide the highest possible taxonomic and phylogenetic resolution. So we may have some very interesting ASVs that behave that display some interesting ecological patterns, even though we are not able to give them a detailed taxonomic names today. But we will be able to easily re-annotate the data, having the data in ASV format. And we will continuously update the data in the bioatlas as reference databases are improving. And having the data in ASV format makes it super easy to do this update compared to having to go back to all of the raw data and remap that to the reference databases. And what type of data are we supporting in SBDI? The sequencing platforms that are compatible with the NF-core AmpliSec denoising pipeline are PacBio and Illumina data. And then currently we are supporting these taxonomic groups and gene markers. So archaea and bacteria using the 16S RNA gene, and then we use the GTDB database. And there we have made a curated version of the GTDB 16S sequence database that where we have removed some spurious sequences. For fungi, we are using the ITS region and the UNITE database, and for eukaryotes broadly, we are using 18S RNA and the PR2 database, and for metazoa, we are soon implementing the CO1 gene and using the BOLD as a database. But other groups and gene markers can be supported upon request. And maybe most important, why should you publish your metabarcoding data in SBDI? Well, first of all, your data can be reused by others. So it's a great way to reusing resources, basically. And you can more easily compare your own data with other metabarcoding data sets, and also with totally different types of data in the bioatlas. And your submitted ASVs will be regularly re-annotated as reference databases are updated. And your submitted data will get a DOI that you can provide in your publication as a complement to the accession number of the raw data in ENA or NCBI. And SBDI data is integrated in GBIF if you opt to do so. So your data will contribute to global knowledge on biodiversity. And then, what are our future plans? Well, first of all, we want to integrate your data in SBDI. So we are now looking forward to receive data from researchers in Sweden to expand the database. And then we will give support for more marker genes, such as the CO1. We will add functionality for searching, retrieving genetic data with the SBDI for our package, and also add more functionality in the actual bioatlas for improved visualization of metabarcoding data. And for more information, go to the ASV portal.biodiversity.se, and you can also contact the support. Thank you very much.